Good afternoon, everybody. Thank you for coming out to the final edition this semester of the Climate Science Center Interdisciplinary Seminar Panels. I want to start off by thanking the Department of Environmental Toxicology for providing the coffee and the cookies, although I must warn you, stay away from the chocolate chips. The chocolate chips are no good. Don't anybody go near the chocolate chips, please. I do want to thank them for sponsoring today. Our panel today is on climate science and invasive species. Please save all mother-in-law jokes until the end. We have three expert panelists today who are here to discuss their research with us, and they'll be able to answer any questions afterwards. First off is Dr. Matthew Barnes. He is an assistant professor in the Department of Natural Resources Management at the College of Agricultural Sciences and Natural Resources. His research is rooted in community ecology, predicting and explaining species distributions and dispersal in aquatic systems. He is particularly interested in forecasting biological invasions, the movement of species beyond their historic native ranges, and conducting research that advances methodologies for the study and management of invasive species and the communities they impact. He utilizes a broad set of tools to address many different types of ecological questions. I'm going to stumble on that word all day today. So his research has included predicting species occurrence with species distribution models and geographic information systems, detecting rare species through environmental DNA surveillance, and investigating species impacts through field observations and laboratory experiments. Ladies and gentlemen, Dr. Matthew Barnes.
Introduction. Thank you very much to the Climate Science Center for the invitation to be here, and thank you all for being here. I'm excited to be here and talk to you about my research on invasive species, and in particular I'm going to focus on detection of invasive species using environmental DNA. And I'm going to get into that in a couple minutes, but because I'm first on the playlist here, I thought I'd start with set the stage here, the nature of the problem that we'll be talking about today. Biological invasions, invasive species. Now what you see here is a video. It's a 24-hour time lapse of all the air traffic across the globe. Each one of those little yellow dots represents an airplane flying around. Okay, this is great. People and products are able to move around the globe further distances and at greater speeds than ever before. But at the same time, all these little yellow dots flying around, all this movement is an opportunity for plants, animals, disease vectors to move around the globe as well. And these organisms that are part of the natural history and the landscape someplace in the world get moved someplace else. These are what we call invasive species. And invasive species have really important environmental and economic impacts. They're among the most important drivers of global biodiversity loss, second only to habitat destruction. And they cost a lot of money, on the order of $120 billion a year in the United States alone. They come in all shapes and sizes. Terrestrial species, the little fire ant, to the vine that ate the salve kudzu. Aquatic species, the little zebra mussel about the size of your thumbnail, to these gigantic 100-pound Asian carp that are jumping out of the water here. And some of them are introduced intentionally, like our tilapia. You might be eating tilapia for dinner some night this week even. While some are accidental, either an accidental release, like the South American apple snail from the aquarium trade, or something like our zebra mussels that came over in ballast water. Now the invasion, it's useful to think about invasions as a process. It starts with species in their native habitat, species where they belong. Those species enter a transport pathway of some sort. Either we collect them because we want to trade them, or they get taken up into a ballast tank accidentally and they're going to hitchhike across the globe. Those species are then transported and released alive someplace new. They establish a self-sustaining population, a reproductive population in the new habitat. and then they spread and cause those environmental and economic impacts that I talked about before. Now, it's useful to think about the biological invasion process this way because each one of those steps represents an opportunity to target specific management actions. We can prevent those species from being taken up into a pathway. We can try and detect and prevent species from becoming established. And even down here where species are spreading, they're here to stay and they're causing environmental impacts. As managers, we can adapt human behavior and try to mitigate those costs, try to bear those costs. You'll see that each one of these steps in the invasion process is represented by a smaller and smaller arrow. And that's because conceptually we can think of each one of these steps as a filter that a species has to go through before it's down here on our bad invasive list. So my research focuses on, as the introduction said, predicting biological invasions, trying to predict species as they move from step to step. And one of the tools that we use to do that is genetic tools. Now, some invasive species, like these Asian carp jumping out of the Mississippi River, it's pretty easy to know where they are, right? You throw your motor through the water, and defensively they jump out of the water. These 100-pound fish are jumping out of the water, trying to get away from that scary motor. But what if we have an environment where the invaders aren't just jumping out in plain sight? What if we still want to know what might be in this environment? What if we've got something like a common carp, not to scale, here in our aquatic environment? Are there, yeah, maybe. Are there other tools that we can use to figure out whether this invasive species is in here? Fortunately for us, species are always shedding material into their environment. Even people, you scratch your arm or you clear your throat, you're shedding a little bit of genetic material into the environment. So these fish, as they swim, they've got mucus coming off of their bodies, water's passing over their gills because they're trying to breathe, and so they're sloughing off some cellular material. They're defecating and urinating into the water. So they're releasing all this genetic material. And so if we know a little bit about the genetics of this organism, the A's, the T's, the C's, and the G's that make up its DNA, its unique DNA code, well, then that sets us up for something I like to refer to as the CSI, or your favorite crime show, environment. We can go out and look for, not the criminal at the crime scene by spotting a cigarette butt and looking for his DNA, but the invasive species, or maybe the endangered species, or some other species of interest in our landscape, by looking for the genetic clues that they leave. We call this environmental DNA. This is the genetic material that organisms are sloughing off into their environment. And in particular, what's novel about this environmental DNA approach is that we don't go out and collect specific sources of DNA. We're not setting out fur traps. We're not taking the time to collect individual scats. Instead, we're taking a soil sample. In my lab, we would probably wear gloves if we did this. We're taking a water sample. We're setting out a dust trap and collecting air. And all of these bulk environmental samples contain a mixture of DNA from organisms. And so if we know the DNA of the particular invasive species, endangered species, other species of interest that we're looking for, we can use standard laboratory techniques of DNA extraction and PCR, things that have been around for decades, to look for clues that our organism was there. Now, a lot of studies over the last 10 or so years have come out and demonstrated that we can do this pretty successfully, detecting American bullfrogs in Paris, detecting Asian carp in the Mississippi River, detecting endangered species as well. What my lab is interested in, my research program focuses on the ecology of this eDNA, this genetic stuff. So after this stuff gets shed from an organism, it interacts with its environment in interesting ways. And I'll point you towards this, this recent review that came out where we set up this framework and really outline this research program. And I'd like to take my last couple minutes here to walk you through this framework, this ecology of eDNA. It starts with the origin of eDNA. We know we're collecting genetic fragments from organisms, but remember I said it might be that stuff off of the gills, it might be feces, it might be mucus. We don't have a great concept of where environmental DNA comes from. And you can imagine this is going to be important if it's from something like reproductive fluids or defecation. We might see little spikes in DNA and then it goes away. But if it's something, if the source of DNA is more commonly something like decomposition or just that mucus sloughing off the body, it might be around in more of a press pattern, around more commensurate, more commonly over time. This has implications for when and how we go out and sample this stuff. Now, related to the origin of eDNA is the state of this stuff in the environment. We don't know, are these really small particles, are these free pieces of DNA floating around the environment? Or is it stuff wrapped up in organelles and cellular material in big chunks of tissue? And that has implications for our understanding of how we collect this stuff. So I'll show you here. If we use if we've got a smattering of eDNA in our environment and we use a really small filter, really small pore size, we're going to get a lot of genetic material from that water sample or that soil sample that we filter. If we use a coarser filter, we might get, I'm standing in front of it, we might get less DNA from that same sample. And if we're trying to relate, say, the amount of DNA we collect to the amount of organisms that are swimming around or crawling around, we're going to draw two really different conclusions just based on how we sample this. Part of this ecology of eDNA framework, I'm also interested in the transport of eDNA. If DNA leaves this decapod crustacean, for example, and floats downstream right into our sample of water that we take, we would amplify that DNA and get what we would say is a true positive. We would detect the species in that environment. But if DNA settles, hangs around for a little while, and then gets transported by, say, wave action long after the crayfish leaves, or if DNA travels in the water column all the way downstream to some other habitat, and we detect it then, that sets us up for a false positive. This isn't really coming from an organism present where we took that sample. And then related to transport is the fate of eDNA. So after DNA is produced, it's in the environment, it's transporting around, also interacting with this is its fate. How long does it stick around? We've got just one organism, and DNA decays in the environment very slowly. That might lead us to one result. But if DNA is degrading really rapidly, a whole bunch of organisms might produce the same amount of DNA in the environment. So we also do experiments where we look at how long this stuff sticks around. Is it commensurate with the presence of the organisms, or are we at risk of collecting relics of organisms past? So when we put all of these together, the origin, state, transport, and fate of DNA, we get the ecology of environmental DNA. Add into that some of the chemistry and technological limitations that I'm not going to get into today. And essentially, we've got my genetic environmental DNA research program up here. Now with that, I'm going to put my contact information up here for you. I'll thank you for your time. I wanted to plug, we've got a workshop coming up at the Ecological Society of America this summer on, again, this ecology of environmental DNA. And I'll finish by saying my interest is in detecting invasive species, maybe endangered species, other large organisms in the environment. But microecologists, hydrologists, genetic forensics experts are all interested in this genetic stuff, extra-organismal genetic material in the environment. And we're going to try and get some of those experts together to share trade secrets and an understanding of this extra-organismal genetic material this summer. So if you're an ecologist, you're going to be there anyway, stop on by. If you're not an ecologist and want to join us, that would be extremely helpful as well. So I thank you very much for your time, and I look forward to the questions.
I was tripping over ecological, much less extra-organism. I'm sure that's a word. Our next panelist is Dr. Samantha Cole. She is an assistant professor in the Department of Natural Resources Management, also at the College of Agricultural Sciences and Natural Resources. She earned her PhD in wildlife science from Texas A&M University, Kingsville in 2010. And she used to, I'll let her go ahead and do that. Studying the invasive brown tree snake, which snakes in trees, much less on a plane. She has worked closely with state, federal, and military organizations in invasive species management and planning projects, and has experience in risk assessment and habitat suitability research for both native and invasive species. She currently specializes in regional planning and management with a focus on wildlife species, which involves conservation planning and assessment of interactions between humans and both native and invasive species at a regional scale. Ladies and gentlemen, Dr. Samantha Cole.
All right, so I just put a few quick things together about some of the projects that I've worked on previously, a couple of things that I'm still working on, and then I'll kind of tell you how those things are sort of coming together in the work that's currently going on in my lab. So this is some of the work that I've done with Texas A&M Kingsville, some things that I'm still working on from this research. I'm glad that Dr. Barnes sort of gave everybody a little bit of an intro to some invasive species ecology sort of information, and talked a little bit about pathways for invasion and those sorts of things. A lot of this research on invasive brown tree snakes was done to assess the pathways through which this species could enter different areas. Okay, so this particular project, I worked very, very closely with U.S. military, very, very closely with USDA, APHIS Wildlife Services, and U.S. Fish and Wildlife Service through, like I said, Texas A&M Kingsville, but have continued this research quite a bit here at Texas Tech as well, asking a few new questions and discussing some different things. So just a really quick background on brown tree snakes. This is Boiga irregularis. They are a very high-profile invasive species. I'm sure most of you have probably heard of them before. They're invasive on the island of Guam. They're native to that particular area of the country, or very close to it, but mostly native to Australia and native to Papua New Guinea and islands in that region. They invaded the island of Guam near the end of World War II, came over on supposedly a military shipment near the end of World War II as a hitchhiker. Apparently, or the data suggest that it was just one individual, potentially just a few or a small handful of individuals that came over from the island of Guam, or from Australia to the island of Guam during that time. And due to the ecology of this species and the ecology of other species on the island and the lack of adaptability of the species on the island to snakes, because there are no other snakes on the island of Guam other than a very, very small 10-inch long blind snake that most people don't even consider a snake. It's usually like that, like more of a worm. So when brown tree snakes came to the island of Guam, they filled this sort of empty niche that no other species on that island was really filling at the time. They're predatory species, and so they were able to prey upon all of the other species there, mostly birds, but also bats and lizards and mammals and all other sorts of reptile species, insects as well. Okay, so they're a generalist predator. So we know quite a bit about the ecology of this species now. They've been very heavily studied since this invasion became known to the world. Okay, so what we didn't know, at least at the start of this study about this species, was their potential to move to other areas. And because Guam is an area that houses a large military base for the United States, we were asking questions, or began asking questions about how the military and transfers and shipments from the military to the continental US and to other areas of the world might enhance the ability of this species to move to other locations, because they are a hitchhiker species. When I say they are a hitchhiker species, I mean they are a known moving species. Okay, so this map sort of exhibits the area of invasion as well as some of the native area for the species. So here we have our Australia and New Guinea, and Guam way up there in the middle of nowhere. So this is just exhibiting that Guam is actually sort of a tourist area for people interested in items and information about World War II. They do actually get a lot of tourists from Japan and from other areas in the Pacific as well. So what I was trying to do with this particular research study is to identify locations within the continental US that receive the most shipments from the island of Guam. Because we have that military base there, we have a lot of household goods transfer from that island. Okay, everybody not only sending mail back to their families, but people transferring from one location to another, one military base to another. So they're transferring whole households worth of items. This species and the ecology of this species is that it is very, very secretive. They're a very long, thin snake species. They're very easily able to hide in very, very tiny areas. On Guam, although most people in the general public don't know it, these snakes live in their houses. They live in their couches, they live in their cars, and their washing machines, and all over the place. Okay, they are there. I can guarantee you that they are there. And most people's homes that I actually went into and helped with some of the searches during my fieldwork time would guarantee to me that they weren't there. And I could go in their house and find 5 or 6 snakes. So they're not difficult to find if you're looking for them, and they are very secretive. Okay, so most people don't even notice them. So when we're transferring large shipments from this island to other locations, there is a very high potential that we could be transferring this invasive species. Okay, there are some known areas in the continental US that have received brown tree snakes via shipment. Some of them have received brown tree snakes alive upon landing. So one brown tree snake actually, well, didn't make it, but made it on a plane, I guess, all the way to Anchorage, Alaska. Didn't survive that trip, but got in the wheel well of a plane, and the plane landed in Alaska, and then they found the snake there. We have found live snakes in places like McAllister, Oklahoma. Once a plane landed there, and opening up the boxes from that particular shipment, live snake was found, or a live snake was found in one of those boxes. So McAllister, Oklahoma, Corpus Christi, Texas, there are areas here that have gotten live brown tree snakes. So what this map shows is all of the locations that receive shipments from the island of Guam over the course of a year. So this large red dot over here is Long Beach, California. They receive almost 3 times as many shipments as the second highest shipping location in the continental US. extreme numbers of shipments from Guam go to Long Beach. And these are multiple large cargo containers worth of household goods. So when you see that the number can actually get above the amount of days in a year, those are the numbers of large cargo shipments that they are receiving, okay? So all of these places get shipments from Guam. Now they may come in different ports, but these are the final locations where those shipments are opened up, okay? So a big part of what I did for this research was doing a habitat suitability assessment, climatic suitability assessment for brown tree snakes, trying to look at, okay, if these shipments were opened in these areas of the continental US, where could these snakes survive? So this is a map of one of the climatic suitability assessments that we did. I'm not allowed to show you all of the research results due to being under contract still for this particular project. But this is one of the early on initial maps looking at potential climatic suitability for this species. So we were able to really sort of parse this out as far as the area where these snakes might have potentially suitable climate and the numbers of shipments being received in all these different areas. That gives us an idea of what's called the propagule pressure for this particular species in any of these zones of the US, which is basically the potential for this invasive to be successful in any of those areas. So this project, like I said, is still going through the contractual process for us to define the high-risk areas, but basically what we're doing is defining these high-risk zones for the continental US, and then trying to get out all of the possible educational materials that we can to those areas. We talk to the port managers, we talk to the military bases in those areas. We try to get them to implement different types of inspections in order to check out all of the shipments that they're receiving from Guam, to inspect for brown tree snakes. We give them rapid response contact information so that we can try to prevent any individuals from moving if they do show up alive, okay? So this is brown tree snake stuff that I have worked on. And my interest in habitat suitability, climatic suitability, and working with all different sorts of species sort of spread from there. And I'm currently working on this other project with the University of Tennessee collaboratively here with Texas Tech and our NRM department on a habitat suitability modeling assessment for a large-scale construction project in eastern Tennessee. And basically the way this came about was that the Tennessee Department of Transportation wants to build a major highway through the middle of the Cherokee National Forest. So I came on as an ecologist with some background information on how to do a lot of this modeling. And basically we are assessing every possible species in that area, both natives and invasives, to try to see how the building of this new development of this highway system might potentially affect those species and the ecology of the area. So we started working on all sorts of different habitat data collection and assessing every possible route of this potential highway that they're going to build through the Cherokee National Forest. And you can see the numbers go up and up. These are just some of the possible routes. This was an early map. They were up to over 37 routes, potential routes now. So we have to assess every single one in order to try to tell them which might be the best route for that particular ecosystem and the way that it affects both native and invasive species in that area. So this area has major problems. This is why they're building this brand new highway. There's already a roadway there, but it's having huge issues with people trying to traverse that roadway. Basically, they have over a 200-foot straight vertical rock wall on one side and a drop down into the Ocoee River on the other side. The Ocoee River is a known whitewater rafting river, so it's very dangerous on a very small 2-lane road that used to be a mining road. Okay. So we're putting together habitat suitability models for all sorts of different species. And again, I apologize, this is the only species that I am allowed to show due to contractual agreements. And surprisingly, it's American black bear, which you would think that they would be more concerned about. But this is basically a similar habitat sort of what we're working on with the brown tree snake. We take all of the ecological data for that species and compare it to what is available on the landscape. Okay, so areas that have darker green, dots spotted through the landscape, those are areas of higher potential habitat suitability. The interesting thing about American black bear is that they have a very widespread habitat suitability because they have such a large home range. Okay, so we also tried to assess what might happen to these species if they were to cross the roadway. So we looked at how those species move and how the roadway might affect them for every potential route of the road. So we did this for native and invasive species in the area. This is another species that I'm allowed to show because it's all over the place. White-footed mouse versus American black bear. And basically this shows us that this particular species has a very high risk area at this zone of the potential new route of the highway compared to how they use habitat in that area. Okay. So ultimately, we end up looking at the potential route, and over that entire route, this is a little hard to see here, every single pixel has sort of some value, some gradient value for how much risk that species is incurring at that particular area of the route. Okay. So this is what our black bear map looks like, and you can tell that there are a few red zones that are high risk zones for that particular species. So basically, sorry, this slide takes a few minutes. We took habitat information, we're taking information about how every species moves, danger of crossing the particular roadway, and trying to identify least impact alignments for all of the species in that area. Basically what we've found so far is that what they have listed as alternative number 4 has the least potential risk for all of the species that we've assessed so far, which is 111 different types of species, everything from large mammals to birds to a few bat species, some endangered snail species, lizards, snakes, and a few extra invasives and things along those lines. So we were all across the board with these models. And that was based upon what U.S. Fish and Wildlife Service, USDA, and TxDOT required us to examine or assess. So we were able to point out where along this roadway they need to do different types of planning in order to try to accommodate certain species or areas along the roadway that they need to do further assessments for the invasives in that particular area and do specific types of management for invasives in those exact zones. So this is a really new and different type of thing. Taking habitat suitability modeling, taking information about the ecology of a species and informing planning, using science to inform planning. It's something that we always talk about doing, that everybody really wants to do, and it's still sort of a new and groundbreaking thing for a lot of organizations. So this is basically how we're working on doing that for a large-scale construction project here in the U.S. Oops, sorry. That's okay.
Thank you, Dr. Cole. I just have one question. Can you tell us if the brown tree snake has been found in Lubbock?
No, it has not been found in Lubbock.
Thank God. I can tell my wife now she won't torch the house.
We don't have enough trees.
Come on. Not you two. My father-in-law does. There's not enough trees around. Sorry, we're not Colorado. Our final panelist today is Dr. Steve Presley. He is professor in the Department of Environmental Toxicology, the Institute of Environmental and Human Health at Texas Tech, and he's the director of the Biological Threat Research Laboratory, which we all need. Before joining the faculty at Texas Tech in 2002, he served on active duty in the U.S. Navy, thank you, sir, as a Medical Service Corps officer, and earned a Master of Military Studies from the U.S. Marine Corps University in Quantico, Virginia. His professional career is focused upon vector-borne infectious diseases with an emphasis on biological threat assessment and countermeasures to protect populations from infectious disease. His operational experiences include the surveillance, prevention, and control of biological threats in the environment, and he has led malaria control operations and research efforts in Africa, Asia, and South America. He has published more than 90 scientific and technical papers, co-edited 2 books, authored and co-authored 11 book chapters in the fields of biological and chemical countermeasures and diseases of wildlife. Ladies and gentlemen, Dr. Steve Presley.
Good afternoon, everybody. It's the first one of these I've attended. I've only got 50 or 60 slides. I've got one. So you may be wondering, what? Can you hear me okay if I'm wandering around? You may be wondering what malaria control operations and public health type things have to do with invasive species. But today, currently, especially with the concerns about Zika virus, they definitely do. I wanted to just provide just kind of a snapshot of what me and my lab group do with the study of emerging and resurgent zoonoses and anthropogenic diseases. Mainly, we look at not at the necessarily the molecular level for the research, or we look more in the transmission dynamics in the environment, how things may become exposed to these pathogens, become infected, how they may move it around in the environment. Currently, we've got, well, actually I came to Texas Tech in 2002, and we started, West Nile virus had not reached this far west yet. So as soon as I got here, having done a lot of that work on the East Coast in the Navy, we started collecting mosquitoes and screening for West Nile virus. We found it and have been collecting mosquitoes, screening them for West Nile virus, St. Louis encephalitis, and some other arthropod-borne pathogens over the years. Got this huge, great database on all the collection parameters for the mosquitoes that transmit West Nile virus. Recently, if you remember last year, there was a big scare, for lack of a better, more academically accurate word, about chikungunya virus being the next, causing the next zombie apocalypse in North America, north of Mexico. Well, it didn't pan out. Zika virus, it's got everybody's attention. For the first month or so, CDC downplayed it as, oh, just another mosquito-borne virus, not that bad. Started getting reports of microcephaly, Guillain-Barré syndrome, effects in people. Now, past few weeks, we found out it's sexually transmitted, and that virus may stay viable and hang around in semen for 90 days or more. Don't know yet. It changed a lot of thinking about diseases or viruses, the arboviruses, the arthropod-borne viruses, like chikungunya, dengue, Zika, how they move around. Because Zika virus was not in the New World until June, or first reported June 2015 in Brazil. That's how rapidly it's exploded and got into the native mosquito population and being transmitted or moved around, transmitted in the environment. The unique thing about these viruses is they're not zoonotic. in the urban cycle, so to speak. They're a mosquito transmission cycle. West Nile virus, as you probably know, is birds, mosquitoes, man, birds. They move around. Humans are incidental or accidental hosts. With the West Nile virus, when it was first introduced into North America from the Old World in 2002 or in 1999, it primarily caused West Nile fever. Since then, until today, that virus has shifted or drifted and primarily causes West Nile neuroinvasive disease. I tell you that because a lot of the same concerns are coming up right now regarding Zika virus, how it may change in the New World species or the New World environs as it establishes itself. Okay, so we get talking on it. So I'll move on. Feral swine. We have a large project, speaking of invasive species, with feral swine. We've got a project with the Noble Foundation in Oklahoma. We had started looking at screening feral swine samples from Crosby County down around Fort Hood several years ago. In Crosby County, we found that 50%, roughly 50% of the feral hogs we sampled, and this was out of a population, we sampled about 40 pigs for that. We found 50% were positive for Francisella tularensis. It was Novacida, it wasn't Tularensis tularensis, but that still told us that feral hogs were moving around a lot of pathogens or could potentially move around pathogens we didn't know about. This project from Noble Foundation, we're screening for, among other things, Brucella species, Trypanosoma cruzi that causes Chagas disease, porcine reproductive respiratory syndrome virus, pseudorabies virus, and Francisella tularensis. In January, out of 32 pigs we sampled, we found 8 that were serologically positive for Trypanosoma cruzi. We're finding large infection rates for pseudorabies and for PRRS. We've got some that we collected in March that we're wrapping up, not ready to report out officially yet, but we're finding Brucella. And there's, if you may or may not know, there was a brucellosis outbreak in humans north of Dallas last year. And so the Noble Foundation, where we're trapping pigs on the Red River, is just north of that. So we're wondering about how that may be moved around the environment. Wrap this all up. Invasive species like feral hogs or like the Zika, dengue, and chikungunya virus vectors, Aedes aegypti and Aedes albopictus, are invasive species of mosquitoes that move in. So I really appreciated your lead in about the flights, the number of flights coming in and out. It's a constant flow. It's a constant potential that I personally believe is not controllable. We're not going to catch it all. We're not going to stop it all just by the volume of invasive species. And if you bend it a little bit and think about these emerging pathogens, like Zika virus is an emerging pathogen. Dengue's not, it's a re-emerging pathogen because we had 42,000 cases in Texas in 1923, but we don't have it anymore. It's coming back. So if you consider a pathogen that's introduced, emerging or resurgent in an area as an invasive species, it all kind of comes together, and it's really just the scale at which you're looking or measuring. So I'm going to leave time for questions. So thank you.
Sounds good. If each of our panels will come down, we'll have you sit here in the middle, and we'll take questions from the audience. Did notice I saw a news story yesterday where the city of Plano, I think it is, has introduced mosquito fish. in their ponds and lakes there to control mosquito population.
Man, you got me on another soapbox. The issue with Zika, and I'm sure most may not know, Aedes aegypti and Aedes albopictus are container breeders. They don't breed in standing water hardly ever. Most Aedes species are floodwater breeders. Specifically, aegypti and albopictus, called stegomyia collectively. They breed in containers. So garbage in the backyard, a kid's dump truck that's left in the sandbox, if it can collect an ounce of water, you'll have mosquitoes breeding in it quickly. Mosquito fish, I mean, unless you put one in every flower pot or vase, they're going to do no good. They're going to be great for St. Louis encephalitis vectors, West Nile virus vectors, Western equine encephalitis virus vectors. But I don't believe they're going to do any good for Zika virus. And that's the big concern right now.
You might get any questions? Yes, sir.
I'd like to start out with a definitional question. How long is something be there before it stops being invasive and becomes indigenous? And when does it get its green card?
We'll build a wall.
I mean, for instance, is a grackle still considered invasive? I mean, I know it's a slippery slope question, but I'm curious.
I don't know that a species ever truly crosses that line between being invasive and actually becoming indigenous to that area. I think it maybe becomes what we would consider a common species for that particular area, but I don't know that it really, at least in my mind, should ever be defined as an indigenous species.
Well, doesn't that then lead to this kind of sort of romantic sense of the Garden of Eden, the original purity of something that's been defiled, and it doesn't get into more policy than actual, because everything's we're invasive.
Invasive species. So I went to a little liberal arts college for my undergrad at Southwestern University and majored in biology, but I minored in sociology. And that really was what led me to study invasive species, because I think there is much of a social human problem and construct as they are a biological thing that we can put our fingers on. And so that's why I think in that diagram that I showed you, the last step there is those ecological and economic harm. And that's going to be defined by people. And so I think I think what you're getting at is that there is a sociological component to it, a human value component to it, and a policy component to it too.
I mean, at some point, the cultural carrying capacity of a species is going to be much higher than maybe it was initially when that species first became a nuisance and recognized as an invasive. As society gets used to having that species around, the interactions with that species and the ecology of the area and the rest of the ecosystem become more regular and more natural. The cultural carrying capacity, the amount of individuals that society will allow and that society can deal with, that carrying capacity will get higher.
Can I comment? Sure. I'm not an ecologist, but I think about feral pigs, feral swine. They've been here a long time, since the mid to late 19th century. Johnson grass that you see growing on all the ditches and fields around here, that was introduced, and it becomes invasive. Feral hogs were introduced and became invasive game hunts. I don't know that. I've never thought about that. When can we quit calling them invasive? Feral hogs, we don't want to, because they are out of control. They found a niche.
Wild horses and burros are pretty bad for the landscape. They're federally protected. They love them.
I'd consider the pigeons at the administration building invasive at this point. You had a question, sir?
In general, do you think the invasiveness of species increases with climate variability or will decrease? Because obviously species evade and home range changes and they migrate, But as we change the variability of our climate more and more, how do we anticipate the invasive characteristics of species? Because obviously, you talk about RNK selection and all those reproductive components of species, they have to come into play also as part of this.
We know for sure that the ranges of invasive species or the potential invasible range is going to change for sure. And so I think that's why this is a really appropriate topic for CSC, right? Biological invasions, global climate change are both really important interacting elements of global change.
That's a tough.
I think adapt and establish is
Yeah.
what's going to happen.
That's a good question because if you look at, for example, wildfire, the common idea is that wildfires can increase. But that's only in the Rockies because that's where all the studies are. Other places might decrease. So it'd be interesting to see a global study of that. Obviously, generalists will be better.
Right, generalists would do better.
Yeah.
Not as well, interesting. One of the reasons I bring this up is there was a paper that was just published about 3 or 4 years ago about one of the largest increasing aspects of invasive species worldwide right now that we don't pay attention to are pathogenic fungi. And that they far outnumber bacteria and viruses almost 10 to 1 in terms of what's happening globally. Part of it's airplane traffic, part of it's just the fact that as climate changes, these organisms are able to adapt more and more to what, and so they're entering new habitats, and so it's and a lot of them are all plant pathogens. We don't pay much attention to invasive aspects of organisms on plants, but we're changing all the time. I mean, sorghum bunt is one of the ones now that everybody's doing.
You mentioned adaptability in your response there. What's really interesting to me about invasions is that back in the 50s and 60s, when Charles Elton, the father of invasion biology, was writing about invasive species, they were interesting because invasions were rare events. Remember, each one of those steps is a funnel. Yet they do so well. We expect that if they're rare events, there should be a really genetically not a diverse population, really crummy population. How are they doing so well? And as we explored more and more, we're finding that actually invasive populations can sometimes be more diverse than the native range populations, because we get things like admixture, where different genotypes from across the range are coming in contact with one another that wouldn't have ever come in contact in the native range. We are seeing those opportunities for adaptation in new niches and these novel environmental systems. And so this adaptability and evolution in invasive populations is a really interesting and important area of research.
Well, I thought that one of the things that we have, obviously in the last 300 years, as human populations have increased and climate variability has increased, what we've done is was like to put those series of traits in all the natural populations, whether we realize it or not, as a consequence of us acting as part of that filter.
Any other questions? Yes, sir.
Do we know the climatic limits for the nasty mosquitoes? Is it the freeze line? Is it?
We do, and we're really above it. But now you're going to be talking about artificial islands that we create in urban areas. You know, on the high plains here in Lubbock, a great example, and I've talked to some folks this morning about, we have trees, we have non-native trees, grasses, shrubs, vegetation, irrigate lawns, flower pots in our backyard. So we've created our ability for Egypti and Albopictus to establish themselves and exist. And I don't know if you've seen the CDC distribution map for Albopictus and Egypti, the most recent one. They did a big survey in January and February of everybody in the country that works with mosquitoes. Said, hey, if you've ever caught them, when did you catch them? Numbers, all of that. Everybody responded back. They published a map last week. The whole southern half of the United States, but it goes into Wisconsin, most of New England, they've been found. And so it's a very adaptive species, particularly Albopictus. For something that was introduced into North America in the 70s, early 70s, in used cars, it's done very well, kind of like
Anybody else got any questions? Yes, sir.
So what do you think with the genetically engineered mosquitoes they had trial with in Brazil and you want
The Oxitec mosquitoes, I think if you're, I'm sure you're all histories or historians on disease control, but you know, the primary screwworm was a major threat on the the southern Mexican border, southern Texas border, post-southern U.S. border at the turn of the 20th century. They sterilized males, released millions and millions of them out of Carpville area, where the head of operations were. They basically eliminated the problem with primary screwworms in livestock. I think the potential for the Oxitec genetically modified mosquitoes is huge if they can produce enough. And one of the problems is primary screwworms were confined and were a regional issue. You know, maybe a couple of million square miles where they occur. When you talk about Egyptine Alcopictus, we're talking about near cosmopolitan distributions. And so getting enough mosquitoes and releasing enough, I think, is going to be tough. But it's very exciting. I mean, for local areas.
You got a question, sir?
I have a question for Dr. Barnes. Like, how do you use the data you generate from the environmental DNA in like the relation to invasive species? Like, there's a big, huge gene pool over there, and like you get a small fraction of the pool from the environmental DNA, and it can be related to some native species itself. So, how do you analyze the data you generate from there?
Right now, the field of people looking for DNA of macrobial species is fairly young, and so we're developing that every day. Right now, we tend to target mitochondrial markers, chloroplast markers, things that are going to be very species diagnostic and differ between species. We don't do, I haven't seen anyone successfully do any population genetics with eDNA because it's this big gross mixture of all sorts of DNA that's in low quantities and low quality. I think that's an exciting frontier if we can figure out how to do it. I think right now some of the big questions that we're trying to figure out are how long does the DNA stick around? Does the amount of DNA in the environment relate to the amount of biomass? What kind of environmental factors in this ecology of eDNA framework influence that amount of DNA, and what kind of DNA is out there? So exciting possibility, but we're definitely not there yet.
So it's more like a metagenomic analysis.
So that's an exciting frontier too. I've got a student that's going to be doing some metagenomics this summer. We're moving from is species X here, our zebra mussels here, our Asian carp here, to saying which species are here, where we take a total environmental DNA sample, amplify using generic plant primers or something like that, a fish family or a group of species, and then sequence them and say which organisms did we find, and that's a really exciting frontier too.
And what about the bacteria present in your sample, because those are so ubiquitous?
This is one of the reasons why we have to bring for that workshop, we're trying to get microbial ecologists and hydrologists and people from diverse backgrounds all in the same room, because of course the microbial people have been doing this, right? You can't culture most bacteria in the lab, so what do? They go out and they look at the essentially environmental DNA. The first person to use the term environmental DNA was a guy named Ogram in 1984, and he was sampling sea vents and the bacterial communities there. So we are building on the shoulders of giants. We need a lot of interdisciplinary collaboration to make sure we're not reinventing the wheel, and to make sure we're combining useful information from all these different fields.
All right, I think we need to go ahead and wrap up today. I want to thank our three panelists. Let's give them another round of applause. That'll be it for this semester. We'll look forward to seeing you all back in the fall. Thanks a lot.